Yep. It's my pleasure today to introduce our speaker, Max Aleman. So Max is originally from South Florida, but decided that he liked the colder climes up here a little bit more. So he came up and did his undergraduate in Franklin and Marshall College, and got his bachelor's there. And then after that, decided to stay again in the state. And so he joined the lab in 2018 to do his PhD work. He has been a recipient of the NSF GRFP, as well as an Northeast SARE proposal as well, which was recently funded. And I think, Max has been here for a little bit. I think everybody who's met him knows that he has no shortage of excitement, and I think that'll come through in his talk today, which is very great. I've been very appreciative. He's joined and started working on actually something in the latter half of what he'll talk about today is something that my lab was not really looking at beforehand. And I think he's made some real progress on it and actually has some results that have a high potential actually of really moving the field forward in a way that we weren't necessarily expecting. So I won't take any more time except for to give you the plaque. So.
Thank you. Okay, let me collect myself. Hola, buenos días y gracias por tenerme aquí hoy. Es un placer. Hello, good morning. Thank you all so much for having me here. It was a pleasure and an honor to be able to talk about something I'm deeply passionate about today, my research. So my only hope as we leave here together today is that I make you all a little bit more excited about aerial surfaces and the bacteria that inhabit it, or maybe the microbes. I guess fungi are important too. So I want to talk a little bit about what I mean by an aerial surface. And so here, when I talk about an aerial surface, it means the outermost layer of the host epidermis, and this typically consists of the outermost layer of host epithelial cells and a protective layer on top of it. And typically we consider this host aerial surface as the interface between the host's interior and the exterior air interface, or air. And so these systems actually exist in a lot of different organisms. You can consider aerial surfaces as our skin or the upper respiratory tract. In fungi, we can see them as mushrooms, or the mushrooms, the sporocarp. And then in plants, we can consider them as the stems, leaves, and flowers. And instead of talking about every possible thing, I want to just focus on mushrooms and leaves for today's talk, for my talk today. And I want to talk a little bit about what are some generalizable functions that we can consider when we're thinking about aerial surfaces. And the first is that the aerial surface typically acts as a protective measure for the host from invasion by microbes. So basically, as an organism attempts to invade into the host, this protective aerial surface will act as a blockade and then cause the accumulation of these organisms at this surface. And this is really interesting because the accumulation of these organisms at this aerial surface allows for consistent exposure to the host, right? It acts as an association between this tissue and the host. And so we also know that at this interface, that the host is actively producing resources within the interior that are being extruded into the interior into the exterior through diffusion. And these resources, or materials as I call them here, are not just carbon or nitrogen or amino acids. There's secondary metabolites that have a slew of functions, from antimicrobial to primary metabolites that aid in the growth and normal maintenance of plants. And so we know that these nutrients will inevitably flow or diffuse from the interior to the exterior, but the intent is to limit how much flows from the interior to the exterior. And we know also that the composition of these resources significantly impacts what aerial surface colonizers are able to persist. This is how we can get different microbial communities associated with these aerial surfaces across organisms in the same area. And so we know that, and up until this point, I'm considering this to be an asymptomatic phenotype, right? Where in this process, this unaltered host metabolic output is releasing resources onto the leaf surface and creating a microbiome that's pretty consistent, right? There's no stressor present, nothing has changed. But what happens when we apply a stressor? And in this case, what happens when disease occurs to the host, right? And this is where my research really stays focused. Sorry, I just wanted to make sure I knew what was next. And so this is where a lot of my work focuses on, because we understand that when a pathogen it manages to infect the plant and is in the host, we know that this causes changes to the host resources within the resources within the host, right? Metabolic changes. And we know that this can alter the microbes that are associated with the host within the host. But what about those associations at the cell, at this aerial surface? And that's where I focus on. So the first kind of question that I have is, how do these surface microbiomes change during a symptomatic host phenotype? So if disease is present on the host, what do those surface microbiomes look like? The second question that I'll talk to you about today is, how do these surface materials, so the resources on the leaf surface, change between an asymptomatic and a symptomatic host phenotype? And then the last one is, does disease improve further pathogen establishment? So if you already have this symptomatic phenotype, does it aid subsequent pathogen establishment? So I want to talk to you a little bit first about this first question, which is, do surface microbiomes change during symptomatic host phenotype? Oh, I don't even know what time. I might be rushing you guys, I'm sorry. I have a tendency to rush. So one way to investigate this that I was really excited was through the bacterial blotch Agaricus bisporus system. So Agaricus bisporus, you may know it as white button mushroom. And these white button mushrooms account for about 85% of all fresh market mushrooms that are sold in the United States. That's a $1.2 billion industry. And what's particularly nefarious about the pathogen that I'm showcasing here, which is bacterial blotch, which you can see as lesions that form on the, let me move away from everyone, which are lesions that form on the mushroom cap that look brownish and lead to this discoloration with like a slicky, slimy feeling. And while they don't necessarily destroy crops or harvests, they will not be able to be sold because no one really wants to eat this. And so, oh yeah, and then over here in the yellow, you can see an asymptomatic. Oh, okay, great. I just wanted to make sure I knew where I was going. So bacterial blotch is particularly interesting because not only is it ubiquitous in these growing houses, because the causal agent of bacterial blotch cannot be pasteurized out of this environment, but another particular interesting problem with bacterial blotch is that it was first reportedly caused by Pseudomonas thalassei. And since then, we know a lot of other Pseudomonas species cause bacterial blotch. But we also know that within these systems, Pseudomonas species are also extraordinarily important for the formation and proper growth of mushrooms. And so within this system, we have a particularly interesting dilemma, right? We know that in order to get down to disease management strategies that are relevant, we have to be able to get these species level resolutions, but in complex communities that can be particularly difficult. And so the first stab at this was done by Samuel Martins in collaboration with the Bull and Hockett Lab. And they managed to get down to the genus level, and they looked at how the composition of the microbes looked like between the symptomatic and asymptomatic communities at different compartments. Just that basically means both at this leaf surface, both at the host's aerial surface, which is right here, this cap, and also the internal tissue. From this point forward, I'm just going to be talking about the communities that were gathered from the associated aerial surface. And this was done by putting mushrooms into water, sonicating them, taking out the mushrooms, and then utilizing those communities. And so this is particular. So the importance of stepping past the genus level when considering disease management strategies, we can kind of orient ourselves here. So let's say we had two communities, and if we got down to the genus level resolution, which we should see right here, but let's pretend we got down these two communities to a genus level resolution. The first problem at a genus resolution is that we may underestimate community diversity. So if we had, for example, Pseudomonads, or these yellow ones right here, right, community one down to species level may only have one species that is dominating, one species of Pseudomonas that's dominating this community, but here we may see two species. And so we are underestimating species diversity or the diversity of these communities. The second problem I've already alluded to is that we actually have to get down to this genus, to this species level resolution in order for us to actually know what members are present and what they might be doing in these communities. And so this is particularly where my work starts. It's really getting down to identifying these taxonomic differences in the Hylia pallis of Agaricus bisporus, which is that aerial surface, and with the associated between asymptomatic and bacterial blotch symptomatic phenotypes. And so as I alluded to earlier, we went into a growing chamber and we collected symptomatic mushrooms across the growing house. And wherever we found a symptomatic mushroom that we were going to collect, we collected an asymptomatic mushroom next to it. And so with these two, they're considered paired samples, asymptomatic and symptomatic. We washed off the microbiomes. Those microbiomes were then extracted for their DNA, and then we submitted those over to sequencing for metagenomic sequencing. The sequences, when returned to me, we aligned, and then utilizing a meta pipeline, and those contigs, we submitted them to Kraken-2 to get down to species level identification. So everything I show you here is the differences between the asymptomatic and symptomatic phenotype at a species level. And so here I'm going to show you our first alpha diversity measure, and it's the observed. And here for a quick just iteration, alpha diversity measures the species evenness and richness within a sample. And when we're looking at here observed, it is the difference in species richness between the symptomatic community, which is here in orange, and my asymptomatic community here in, I don't know, like light blue, teal. Okay, so what we can see is that we do observe significant differences in the species richness. So there are differences in the species that are present between our symptomatic and asymptomatic phenotypes. And this is further corroborated when we look at CHA01, CHA1, right, which leverages richness instead of evenness. And what that means is it will try to compensate for rarer species being present in your samples that may not be necessarily evenly distributed within that community. And so this we also see as being significant. But when we look at richness or the inverse Simpson, which what it does is weighs how evenly distributed are the species that are present within your sample in that sample, we see that there is no significance. And this is interesting because taken together with these three, this means that enrichment of rare species in the symptomatic communities is what's driving a large portion of the differences observed between phenotypes. So taken back, right, there's species that are being enriched in our symptomatic community that are not necessarily present in our asymptomatic communities, but they're not being enriched to or they're not present in our samples at a level that's technically significant. Go ahead.
Did you look at Shannon as well, and did you see the same thing?
Yes, we did.
And the same thing as inverse Simpson?
Yes, we did. Yes, we did. And I have that data. So I can answer that question without without mitigating it in a moment. My likely is that the Simpson is significant and that the inverse Simpson was not significant. That's my gut check feeling. We tossed that, the reason we didn't present that data is because it continued to reiterate the same story. And we'll show you now next when we look at our beta diversity, right, that we see that there's not a lot of separation in our communities. And so let me align you. Our beta diversity is the ratio between, so it just measures the distance or dissimilarity between our samples. Our teal color again is our asymptomatic samples. And then our orange here is our symptomatic samples. What we can kind of initially see is that for the vast majority of our samples, there's no separation between asymptomatic and symptomatic phenotypes. But interestingly, to me at least, I think that there's three distinct groupings of asymptomatic communities when I look at this, right? And this immediately made me question, what is driving the differences between these asymptomatic communities? And I'll point out that these two up here, this grouping here, and then this grouping here, are the three kind of asymptomatic groupings that I'm talking about. And I was just curious if we could see any differences. And so we can take that by utilizing an unrestricted approach. And an unrestricted approach allows us to say, okay, this is all the same phenotype, but I'm curious if within my phenotype, there are samples with species that are enriched, that are different from the rest of that phenotype. So we can pull out within the same phenotype samples that are kind of subgrouping with each other. And in this case, we were able to find a group of significantly enriched species that were driving this. Before I get there, I will let you all know that my initial thought when I assessed this data was that this community would look very similar to each other and that we would expect to see that these are symptomatic for disease, rather that they have high relative abundance of the pathogen or a pathogen, and that's what's kind of causing them to look similar to the symptomatic communities. I was wrong. I was like flat out wrong. What's actually driving a significant portion of the differences between this grouping of symptomatic community or asymptomatic communities and this grouping of asymptomatic communities is actually Pseudomonas thalassai. So as I said earlier, Pseudomonas thalassai is the causal agent, or typically prescribed causal agent to bacterial blotch. And so for what we consider asymptomatic or showing no lesion, thalassae is making up between 50 to 80% of the total reads that are attributed to those samples. So overwhelmingly, thalassae is dominant in these samples, and yet these are still identified as asymptomatic, which is particularly interesting. But what I thought was also interesting associated with this is that the samples that had significantly increased total abundance of Pseudomonas thalassai also had significantly enriched Mycetocola SPJXN-3. And this is another known pathogen of mushrooms, but typically of the stipe, which is like the little bottom portion, more like the stem of mushrooms. Don't yell at me. But another interesting point to be made here is that overall in our symptomatic communities, we had Mycetocola enriched past 0, so about 1% or higher it was present in our communities, but we saw kind of a sheer distinction between the communities that were 0 or very, very low and the communities that had these very high Mycetocola in our asymptomatic communities. And lastly, to touch on those two right here at the top, what seemed to be driving most of the differences between these two samples and the rest of our samples seems to be Pseudomonas putida. So putida seems to be between 33 and 45%, excuse me, it's 33% and 45% of the total composition and are overwhelmingly dominant in these communities. Okay, interesting. So then what are the differences between the phenotypes? So now we know within our phenotypes what's happening and what's maybe drawing some of these differences, but what about between these phenotypes? I'll tell you, there's, excuse me. And so we utilize the GAM model for this. And what a GAM model allows us to do is it simply takes into account all of the species that are present across all of your samples, and then it compares by phenotype. So if you have significant enrichment of species X, here indicated by P, in a subset of both your asymptomatic and your symptomatic samples, if more was enriched in your symptomatic, or there's a greater abundance of the species in your symptomatic samples, this model would say that it was significantly enriched in your symptomatic samples, regardless of if your subset in your asymptomatic samples were somewhat enriched. So what we come back here is that there's only about 13 species that when we have this kind of restriction on are able to differentiate between all of the symptomatic communities, and all of the asymptomatic communities. And most of these, as interestingly enough, are Pseudomonas. And so this particularly is where we kind of, yeah, okay, great. Okay, and so when we take this, so these are all the species that were identified as significantly different between our symptomatic and asymptomatic. If we go ahead and we look at that on our total abundances, on here on the left, you see the species that previously we identified as being significantly enriched. And then on our x-axis is the relative abundance. What we can see is that for the most part, that the species that were identified as being significantly different seem to be asymmetrically enriched. And so this was particularly important for us because previously to this data, we've been getting a lot of significantly enriched in only a couple samples. And what's really nice about this particular data is that it gives us a set of 15 species that we can further investigate that we know are either enriched in our symptomatic or our asymptomatic, and not in a subset of our samples in either one. Okay, so kind of to conclude this section, great. To conclude this section, we had the goal of identifying these taxonomic differences in that mushroom surface. And we can see on our first one is that there is an increased species richness in our caps and the symptomatic for bacterial blotch, even if no difference in that species diversity technically exists, right? Because that's not very, because of the similarity in the evenness. And our second takeaway here is that Pseudomonas species were significantly enriched in asymptomatic communities. But if those asymptomatic communities are on their way to becoming symptomatic, or if they're actually showcasing a asymptomatic phenotype, even with a huge pathogen population, that we cannot have determined here. And that is something for future work to kind of disentangle and establish. And so I actually do have future work here, and it's functional annotation, which is very imminent. And this is to kind of filter back to our resources or the capacity to utilize resources changing in association with these microbes or in these communities. So does disease cause functional differences in these microbial communities? And I don't have that data to share with you all today. That is where we're going in the future. Okay, so to come back to the kind of overarching figure. So we've just chatted about do surface microbiomes change, and we look at that in the mushroom pathosystem, or the Agaricus sporus bacterial blotch pathosystem. And now I want to transition a little bit to talking about do these surface materials change during a symptomatic host phenotype. And I'm going to transition from mushrooms now into the tomato bacterial blotch, bacterial speck, excuse me, system. That's out of order. Okay, and so the bacterial speck is a pathogen, is a disease of tomato that is characterized by these small lesions, the small necrotic lesions that typically have haloing around them. And it is a very devastating pathogen for tomato. And if this pathogen is present in a subset of tomatoes in your field, it can be very quickly very devastating. And so I was very interested in this because we know that this pathogen alters the host's metabolism for subsequent invasion. And this is particularly interesting for the question, which is, does altered metabolism lead to changes at a leaf surface, metabolite? So I have two goals now. The first goal is to determine if we inoculate a leaf with a pathogen and we see diseases, do we see changes to the leaf surface metabolites that are associated with the site of infection, as well as the leaf closest to it? And the intention here is, do we see differences only localized, so near where the pathogen is already infecting, or do we see a systemic change in the host's leaf surface metabolome? And so my second goal is to determine if the changes in the leaf surface metabolite profiles affect associated microbes. And so we started off, I started with a relatively straightforward approach. So step one is just the simple application of our pathogen, or in this case, I say defense response elicitor, into what we consider a treated leaf. 72 hours post-inoculation, I collect both this leaf, which is the treated leaf, and then the leaf that's next oldest directly diagonal to it. So this leaf right here. We collect both of these leaves and we put them in 70-30% MEOH H2O, and then we extract the leaf surface, those leaf surface metabolites, and then you can dry them down. And I will say that when I initially started this protocol about 1.5 years ago, the process took me a month. From extraction of leaf surface metabolites to fully drying down and then being able to reconstitute them, it took me about a month. Now it takes me about a day. So we can, this throughput is actually very efficient, and you can go through a lot of samples very quickly. So once we had these metabolites in our aqueous sample and we dried them down, we then had the interesting question, which is, how do we observe them? UHPLC is extraordinarily expensive. For anyone that has done it, you will know. For anyone that hasn't, metabolomic data is expensive. So we wanted to, since we were doing a foray into do these metabolites change on the leaf surface, we wanted to utilize a different approach. And this particular approach was by utilizing nuclear magnetic resonancing. And this simply produces this graph from our samples. And so what we have here is just peaks positions, and each of these peaks that you see here correspond to a hydrogen in our sample, a different hydrogen in our sample. And these hydrogens can give us what I like to call a fingerprint. So it gives us a fingerprint of the relative hydrogens that are on that surface, or the leaf surface that we extracted. And that is a proxy for the metabolites that are on the leaf surface, because you won't have that same position unless that hydrogen is interacting with the chemistry in your samples identically, right? Or in that same molecule. So what I'm going to show you, no, actually, not just yet. Okay, so from here, when we took our samples, we can get these kind of peaks. And when we get these peaks generated from our samples, we can actually go down and break them down by their region into likely or most likely function. And this was done because previous research has already gone through and corroborated NMR peaks in tomatoes specifically with their associated chemicals or their associated molecules. So we can go through the peaks that I have generated, and I can pull out these peaks and then create an analysis for you. And I'll show you that in a moment, but I want to show you first the peaks that we generated, because I think they're actually really, like, I don't know, they're really cool for me. So I'm going to show you peaks. And what you're going to see is on top is going to be control. On the bottom is going to be my DC3K treated leaves. So, or metabolites from leaves that were either, from plants rather, that were either inoculated with DC3K or their associated leaves right above it. And those are called distal. And so we'll go right here. So as I said earlier, we see control. We see that P. syringate PV tomato, and treated just means where we directly applied the elicitor. And this black box is where I want your eyes to go to. And so what we're looking at here is, if we go back, this is the sugar region. So I've just blown up in my samples where the sugar region is. And I want you to just focus right here, because what we see is that there are differentiations in peak structure, right? And the presence of peaks, but overall, we also see a difference in the intensity of these peaks, which means that those molecules there were lower, or rather the amount of hydrogen present in these peaks, or these samples of these peaks, was lower. Now, I will say, I'm looking at sugar on the treated leaf of a pathogen in comparison to a control leaf that was just with buffer. So we should all have healthy skepticism on how different this is. So let's look at the distal. And in the distal, we see a really interesting phenomena, which is, so same premise, right? The control DC3K, this is the peak graph. And here what we're seeing is that there are differences in the topology of these peaks, right? Because this is a double, right? This is a single, this is there's a little bit of topology differences, and we can see that topology differences here. But what is more important here is the intensity differences. This was particularly interesting to me because it's the first time when we saw some kind of evidence that there may be more sugars, there may be something more present in the distal that may aid pathogen establishment. And so after this, I was like, okay, what about phenolics? Well, phenolics, there are differences that we don't need to squint at. So here we're again looking at control. This is the pseudomonas, the DC3K, and then we're looking at the leaves that were either applied with buffer or DC3K. And here we see a huge expression of phenolics, and this phenolic profile is very different than control. And I think it's okay to be skeptical and say, well, maybe that's just the pathogen for doing that. Well, when we also look at the distal leaf, we see differences in these phenolics, right? And that in this case, it's not an organism utilizing it, or at least our pathogen utilizing it. These are hopefully, hypothetically, stimulated by our pathogen. And so we can see that this occurs all across this graph. And each of these are differences in hydrogen, which putatively mean differences in the metabolites. So we can, what we can do then is we can take these peaks, right? And then we can align them against each other to compare if these peaks are present across samples. What we find is that we do have, so we're looking at a PCA spore plot, and what we're finding is that our samples are different, right? And specifically, this DC3K distal leaves, the metabolites that are present are very different and significantly different than the types of metabolites that are present in any of our other treatments. And this is probably the first time that I got some really exciting news to me, because this is the first time where I was able to finally differentiate between samples that were not treated with my pathogen, which would be right here. And even though we don't have separation, I'm very excited with how similar these samples are, because I feel that once we step away from NMR and go into UHPLC, that we should see a lot more differences between our control and our treated with a pathogen. Give me 1 second, I just want to know where we're going here. Okay, great. Okay, and so this was particularly exciting and gave us a really interesting outcome, which is that there is a significant pathogen effect on the changes in the leaf surface metabolite profiles. But at the current resolution, which is NMR, it's impossible to really be able to fully differentiate down to the compound level. It's just, I tried, it is impossible. So that was really exciting. And so our second goal of this section is to determine if those changes that we did observe affected the metabolites, or excuse me, affected the associated microbes. And instead of doing this at an entire community level, I became very specific with this. I saw sugars and my immediate thought was, does that just help everyone? And so in order to do this, I set up an in vitro experiment, which I'll talk more, but I wanted to incorporate a pathogen and a growth promoter. And the intention here was to differentiate if the changes that we observed of those metabolites on the leaf surface, if they aided all organisms that were present in the community, or if it would just aid our pathogen. And so we did this by taking those treated leaf surface metabolites, those distal leaf surface metabolites, and then we diluted them in M9 with no carbon. And the intention here was to see, in just growing with those extracted metabolites, can the pathogen or our growth promoter grow in our samples? And so we used, I will be talking about both DC3K and A506. Here, I'm going to be showing you a growth curve, right? And so this is OD600 on the y-axis, time is on the x-axis, and this is growth of DC3K in either control-treated metabolites, the distal control metabolites, DC3K treated metabolites, or the DC3K distal metabolites that were extracted from those leaves. And what we can see is that overall, more often than not, our pathogen does best in the metabolites that were extracted from leaf surface, from the distal leaf surfaces. This was really exciting to me, right? Because this is this is an N of 15, and we were able to see this consistently every time we did it. And we also did this with 3 different leaves. So we extracted metabolites from 3 different experimental treatments of these, and then did this an N of 15 with the biological differences of 3. And we still managed to get those results, which was extraordinarily exciting for me. I know that everyone's like, that was confusing. And so we did the same thing with A506. And what was also interesting is that we also saw that increase in the, so same set of treatments, control treated, control distal. We have the DC3K treated, and then the DC3K distal. And so this is growing A506 in these leaf surface metabolite profiles. And what we can see is that DC3K treated and distal seems to improve A506 growth, right? So it seems like the metabolites that are changing at that leaf surface may preferentially aid other organisms that are present. And so determining these changes in leaf surface metabolite profiles affect microbes, we can say with some certainty that it seems like that there are changes observed, that the changes that we observed in goal 1 seem to have an effect on both DC3K and A506 in vitro, and these can be attributed to the leaf surface metabolite profiles. Okay. I'm rushing. I always do this. Sorry, everyone. I get so excited to talk and realize that I'm like rushing everyone. So I want to talk about maybe my last goal here. And this was to determine if initial infection of a pathogen on a plant improves the subsequent pathogen establishment into another non-infected leaf. So if we have an infection on that treated leaf, will we see improved pathogen establishment on the distal leaf of the same plant? And so that is the experiment we set up. So we applied our either the pathogen or A506 on our treated leaf 72 hours after application. We would add either DC3K or A506 on the treated leaf. And then 72 hours post-application at the treated leaf, we would take the treated leaf and then do a CFU on rifampicin to identify if we see any differences in the CFUs that were grown in the distal leaf. And so the way that we did this is by calling it a phase 1, phase 2. And you'll see from here on out this kind of null sign, which means that the treated was with a buffer, and then we applied A506 after, and that kind of follows the same way. So we applied DC3K first, and then A506, and subsequently. And so I'm going to first show you our results in this experiment with A506 and DC3K. And what we found was that there was, oh, I forgot. Okay, sorry everyone. There's going to be something wrong on the next slide. I got you though. Okay, so what we did see is that, so we replicated this experiment 30 times. Right, so we were able to quantify CFUs 30 times, and on average, we see that there's no difference in the growth of A506 between the control and when we apply the pathogen first and then attempt to grow A506 in the distal leaf. But that is not the same that we see when we do this with DC3K, which is really exciting. The thing that I'm missing here is that these are all significantly different from each other, right? This is statistically significant from both of these, and this population is significantly different from both of those. And I'll tell you more as we go. So here, we're still looking at log 10 CFUs, and we're looking at here in the gray box, this is when we apply the buffer to our treated leaf, and then DC3K into our distal leaf, A506 in our treated leaf, and then DC3K on our distal leaf, and then DC3K on our treated leaf, and DC3K on our distal leaf. And what we find is that on average, DC3K is growing about threefold greater than if you have DC3K just as initial colonizer. So the first initial infection, the secondary infection is about threefold greater, or the population it's able to establish is about threefold greater. And that we also see an effect by A506 is particularly interesting to me. This is something that we hadn't seen before. And so this gets to an idea that there's possibly susceptibility, like induced susceptibility that's occurring between when our pathogen is infecting the lower leaf and then the inevitable changes on the upper or distal leaf. And this is really exciting because this means that if a plant is infected and the pathogen is able to manipulate the host to such and to an extent to alter the host's metabolites, it can be preferential. Some of these changes can preferentially affect the pathogen and subsequently aid it in establishing larger populations further on within the same plant. Okay, so to kind of recap, our first question was if these surface microbiomes change during symptomatic host phenotypes, and it seems that they do. And we see this because there are species that are differentially enriched when compared between the symptomatic and asymptomatic mushroom caps. 2, do surface materials change during symptomatic host phenotype? It seems that they do. And it's exciting for me that these researchers at the leaf surface seem to alter in a manner that's dependent on the presence of the pathogen, or the disease rather. And further than that, it is extraordinarily interesting to me that these changes are going all the way to an uninfected tissue, or that distal tissue. That's fascinating to me. And so I think that this, in my opinion, has implications for host surface associated microbes or microbiomes, right? As well as host tolerance to subsequent pathogen infections. And then lastly, does disease improve further pathogen establishment? It seems that it does, because we observed that the pathogen populations were typically, as I said, threefold greater on the distal leaves of DC3K than in either the uninfected or in A506 treated first. And this is extraordinarily exciting because in the presence of A506, we see that DC3K improves its growth, but that relationship was not reciprocal. And that is fascinating to me, and somewhere that I wish to be able to explore more one day. And with that, I'd like to thank everyone for being here. I am extraordinarily grateful to have had this opportunity to share my research with you all. I'm extraordinarily grateful for my lab for giving me this opportunity to just explore a really interesting question. I'd like to thank my funding agencies. And with that, I'd like to ask, do you guys have any questions? So sorry, I like.
As per usual, I guess the first question to come from a.
Hi, yeah.
Awesome job, Max. Looking at your second objective, I was curious what your hypothesis was on why the metabolites extracted from your control treated leaf added to growth versus the controlled distal.
Yeah, so yeah, absolutely. Couple things that happen there. The first is we use 3 to 4 week old plants, and typically, right, what we do is we'll set out we'll grow out a bunch, and then we'll pick the ones that are all the most similar in growth stages. So they have like 2 leaves that look the most similar, and then we'll let them grow for 1 more week. Oftentimes, they all have very similar, and we can corroborate this and we can control this by normalization, right? But inevitably, typically, those distal leaves just seem to have less metabolites. It could be that we either are getting them as they're still fully growing, right? We typically get them once they seem to be fully matured and have an already other leaf growing. But in that time, there's some ontogenetic differences that could be playing a part, right? But what it may be, though, is that we're overcoming those ontogenetic differences when we have a pathogen elicitor.
Okay.
Which is interesting, which I don't know.
So as a follow-up, typically do younger leaves have more metabolites than older?
Typically it's the other way, right? Typically,
Okay.
that's the expectation because the younger leaf is acting as a sink, and so it's taking in a ton of resources, right? Versus these mature leaves, which is why we're utilizing them, we expect them to be already fully exhibiting their metabolic profile, and that should be much more robust than in a non-mature leaf
Interesting.
or an immature leaf.
Thank you.
Absolutely.
I was just wondering how like these diseases, like the blotches are like spreading between mushrooms
Yeah.
or how like the disease is spreading between like tomato leaves. How does that?
Yeah, absolutely. Great questions. So let's start from tomato leaves, right? The way in which bacterial speck moves around is it will act as an epiphyte on the leaf surface. So it'll persist on the leaf surface until a rain event occurs. And then rain will aid its mobility into the leaf through the stomata. Once that occurs, then you have subsequent infection through other rain events into the rest of the leaf and then across your field. And when we look at blotch, I should have blotch over here. When we look at blotch, the pathogen is actually already present within the system all the time. It's almost ubiquitously present within these growing systems. And what actually initiates pathogenesis, or from the transition into what it seems like an asymptomatic into a kind of diseased symptomology, is the presence of water. So water left standing on the mushroom cap within like 30 minutes leads to like an insane increase in the pathogen's population, which is a good precursor to disease. Yeah, go.
So I was curious about your community differences when you had some of your asymptomatic ones that have this pathogen at high levels. So I'm curious if there is potentially now, so, okay, it was important to get down to the species level differences. What about strain level differences?
Yeah, crazy, great question. Yeah, oh, absolutely. We need to get down to them. And we're the only thing that limits us getting down to to the species or subspecies level resolution is realistically databases and how much you trust your databases. So one thing that is inevitably going to happen with this data set and between the collaborators of this work is Dr. Bull, I believe, has a robust library of pseudomonads that her lab is continuously developing, and utilizing those already informed strains that we know are correct to better perform the.
I get it.
Yeah.
So also though,
Mhm.
you don't necessarily, to answer my question for this, need to get the correct species identification. If you know anything about what are the virulence factors in the pathogen.
Yeah.
You could look for whether those are present or not.
Yeah, which is inevitably the intention. Well, as we take this a step further, we have this, we're already doing MAGD replication for these samples with the intention of not only getting biosynthetic pathways later on, that is not my work, but biosynthetic pathways, but also other subsequent functional differences that are either enriched or are just different in our communities. It is in the pipeline.
I'm just looking for to last work for that.
Yeah, let me do that. I'm gonna add that as a little note.
I have like a few different questions.
Go for it.
Okay, so I'm actually very surprised by your last result.
Yeah.
Because the way I think about like plant immunity is
Mhm.
that when you have an infection in one leaf, the distal leaf, that would increase basal levels of defense, and then the distal leaf would be less likely to be infected, right? Like that's if it's working correctly, that's what's happening.
Yeah.
So have you done or are planning to do any RNA-seq of the host?
No, so RNA-seq is a little bit outside of my expertise. We have qPCR, which allows us to identify that there is elicitation of our expected pathways. But to get to a little bit more of your point, I think this is a very subsetted samples of the work that I have. I have it for jasmonic acid, salicylic acid, and mutants. Because what is not in this conversation, because it's a story that I have to be concise about, is that coronatine and there are virulence factors secreted by the pathogen that manipulate the host metabolism, specifically two. the type 3 secretion factor, type 3 secretion system, and carotene. Carotene specifically is a methylgesmonic hormone mimic that we expect to possibly be aiding in that signaling. And we know that carotene, which you guys don't know, I know, right? Now in my work, I know that carotene is actually driving different patterns of metabolites on leaf surfaces. So if we remove carotene from the pathogen, the metabolites on leaf surfaces are actually different.
Cool.
Yeah.
That's a nice story for that molecule. Then the other thing I want to comment is every time I see like 18-hour growth curve where you sampled hourly, I
Yeah.
was like, that probably meant you slept in the lab. I've done that, so awesome job.
Yeah.
And then my other question is for the mushroom work.
Yeah.
With the beta diversity plot where you're looking at the asymptomatic, symptomatic.
Mhm.
So I see there that you just did an unconstrained ordination, but have you tried doing a constrained ordination? Because I think you'd probably be able to pull those differences apart.
No, I have not. I did not try to.
I had a similar suggestion to try to put loadings on your plots and see who's pulling your points each direction.
Right.
Yeah. I can even send you some code for that if you want.
Yeah,
That would be awesome.
because like with that story there, I'm pretty sure you'd have that pathogen on your loadings pop up over there. I don't-
Oh, yeah.
So.
It's cool.
Yeah.
Could you go to the list of the Pseudomonas that we have in these?
Your slides are beautiful, by the way.
Thank you. So it's likely that Pseudomonas zotoformans may be one of the pathogens as well.
Huh. That's interesting. So when I saw mycetocolon, that's when I started going like, oh, I need to actually go through now and like look at each of these and see what literature has already established there for them.
I could, like you said, if you want genomes or whatnot, I could tell you which ones are closest to that from Pennsylvania.
Sure. So that may be something that like in collaboration between the three of us, that we may pull out if we have already that easily accessible.
Anyone else?
Good question. So with your shotgun data, you classified your reads in the Kraken. I was wondering what proportion of your reads remain unclassified just because of limitations of database?
Yeah, so from the limitations with Kraken's database, we only had on which I should have I'm gonna have a slide on that. Thank you. I think it was only like on average about 3% of our reads were not considered aligned or identified down to a species level. Only about 3%. Great question though, and that will be on a slide so that I can attest to that. That's an important factor, but I believe about 3%. And this was done, like as I said, we did this through the MetaRep pipeline, which aided a lot of those constraints.
Anyone else? All right, let's thank, one more time.